Hi everyone, my name is Will DePas. I'm an assistant professor in the Department of Pediatrics. I'm going to tell you in this little video today about the basic overview of the work that we do in the lab. So we're bacteriologists. We mostly study the chronic pulmonary infections that occur in cystic fibrosis patients. Our main focus is on biofilm formation and specifically in non-tuberculous mycobacteria, which are emerging pathogens. So bacteria can form biofilms or multicellular communities in many different environments. This little slide, I'm just illustrating that in the non-host environment, so anything outside of the human body, and then in the host environment. So opportunistic pathogens like NTM, right, normally exist outside of the host. When they get the chance, they can inoculate or infect a susceptible individual, such as a patient with CF. And then in the context of disease, in the site of infection, they can form these multicellular communities called biofilms. They can also usually do this outside of the host, but importantly, it tends to be in response to different environmental cues. And they are sometimes different types of biofilms, right? So different matrix components, different regulational pathways that are involved. So we're really interested in studying biofilm formation in the actual context of infection and disease. And we really want to understand this because biofilms tend to confer resistance. So in the infection, resistance to things like the host immune system, phagocytosis, and antibiotics. Outside the host, things like disinfectants or predation, right? So it's important if we want to control any given bacterial community to be able to control this biofilm formation response. We use a three-pronged approach to study biofilm formation in the lab. On the lower left here in this blue box, you have your in vitro studies. So this is basic lab assays that you have set up so you can really understand the fundamental, the mechanistic properties of biofilm formation. So these are some tractable assays you have where you can alter environmental cues and get biofilms to form, get them to disperse, and try to understand that transition process. So with mycobacteria, this is relatively straightforward in the lab. What we have is a little liquid assay. So if you grow any culture of mycobacteria, usually they spontaneously clump into these aggregates. So this is our biofilm state. If you leave them long enough or alter sort of the chemical conditions of the medium, you can get them to disperse into a planktonic population. So we're really interested sort of just in using this basic system really to get at the nuts and bolts, the fundamentals of this process to understand the basic mechanistic properties there. And then on the opposite end of the spectrum, what we want to do is characterize biofilms in situ, and that by that I mean in the actual tissue that's infected. And to do this, we need some good imaging technology, and that's what we use. And our real goal here is to quantify things like biofilm size, biofilm morphology, interactions between biofilms and host cells so that we can really get a good target to design our in vitro assays around. We can know what types of biofilms we're really after. And this is just a general overview of that imaging technique that we use. We call this MyPact HCR. It involves taking a tissue sample. In the case of CF, this is usually expectorated sputum. You fix it in formaldehyde, embed it in a acrylamide matrix. This provides some structural support. And then you can optically clear it by removing a lot of the lipids. And this just makes a now stable, transparent tissue that you can do three-dimensional imaging through. So light goes straight in, light goes straight out. So then if you label your bacterial pathogens and a few host components or host cells, you can then do 3D imaging and get a good sense of the biogeography of the infection setting. So things like you can quantify biofilm formation, any kind of spatial parameter that you want. And this is just a few pictures that we can apply this to different systems. Again, CF sputum is the thing we're mostly interested in, particularly with NTM. You can see a few different bacterial pathogens here, one in magenta, one in green. So they're forming, you can see biofilms, and then we can start to characterize that in situ community as best we can. So it's really the go-between then of the in vitro basic studies and the in situ characterization that we're interested in is what kind of biofilms form during infection, how can we take those biofilms apart in the lab. So then before I sign off, I just want to introduce you to the lab members. We have a second year PMI student, Mitch Meyer, undergrad scholar Carlos Rodriguez, and then my technician from Alex. So thanks for your attention, and yeah.